you didn't have enough from yesterday. But hopefully these will be complementary to what you learned about yesterday. And we'll have presentations by both by myself and by a number of members of my lab who have prepared some presentations and demos and tutorials for you guys. And we may not get to all of them depending on how the timing goes, but this is sort of the generic plan. First, I will give this intro overview talk about what is rule-based modeling, why do we want to use it to specify biochemical networks. I was going to give a short demonstration in here, but up here in the classroom where you guys just watch what I do. Then we'll go into the lab and do some example models. We could have another discussion about modeling methods before we get into the hands-on stuff, if you'd like. Jihan is going to go through an extended example of modeling the CAMK2 system in BioNetGen and getting into some of the gory biochemical details that and where you really see combinatorial explosion in action. And then Sanjana will present some of the tools that we've been developing to visualize large rule-based models and to calibrate models to data. So this is really parameter estimation. How do we, what's the workflow for doing that with typical rule-based models? Really, it's not specific. The methods that we're using here are not specific to rule-based models. They would pertain to any biochemical kinetic models, and they could even be used with MCell if you had a big enough computer. And then, time permitting, we'll talk about how you could get models from the literature into BioNetGen, RuleBender, and do simulations and so forth, and visualize models. So lots of things are possible, and we'll see how far we get with that. Yeah.
What do you mean by a big enough computer? Like, what is a big enough computer?
Well, so the methods that we've developed so far for calibrating models to data are parallel, so we can run 4 to 8 processes at the same time with the methods that we're using there. So if you have a cluster that has some pretty strong nodes, and you could use, say, 6 to 8 of them, that would be optimal for that method. Now, if you have a big M, not necessarily. It depends on the model. So, but for an MCell model, that would be a very slow process because each simulation might take, minutes to hours. Most of the biochemical kinetic models, the simulations will take seconds to minutes. So it's feasible to simulate the model 100,000 times or 1 million times in the course of a couple of days. It's not feasible to do that with most sort of non-trivial MCell models currently. So there you would want to be able to run hundreds or thousands of processes in parallel at the same time. And there are parameter estimation methods for doing that, but we haven't, and that even work with rule-based models as well. Those tools aren't being developed by us at the current time, so it's not something that we develop internally. So broadly speaking, what we're trying to do here is to model cell decision processes, the biochemical networks inside cells that process information and lead to different changes in cell fate. It could be, and this really could represent any kind of biochemistry. Here we have signaling through growth factor receptors coupled to GPCR kind of receptors. But we have many general problems where we want to model networks, and we want to develop predictive models of those, in order to really understand mechanisms, and ultimately to change how those networks operate. And many of these networks are involved in signaling that's causing cancer, for example, or signaling in the immune system, which is an area that I focus on a fair bit, where the kind of immune response you mount is determined by the kinds of signals that a cell receives, and different signals will result in different outcomes. And how do we modulate the behavior to suit a particular desired outcome and to ultimately to treat a different disease. Somehow this slide got a little bit messed up. So it says that we use an approach called rule-based modeling to build and simulate models. I'll talk about what rule-based modeling is, and BioNetGen is the software that we develop that carries out the rule-based simulations of rule-based models and manipulates them. And RuleBender is the interface that we use to interact with BioNetGen. So generally speaking, this is a multi-scale problem. We have at the lowest level of resolution complex biomolecules such as proteins. They have a complex three-dimensional structure. They're composed of they're a polymer composed of amino acids that have very specific chemical and biophysical interactions with other molecules. And one of the fundamental properties of these proteins is that they're not just simple nodes in a network that carry information from one node to the next, but they're actually complex, multifunctional machines that can interact with many other molecules in the network. And we know that manipulation, that binding or mutation of a single amino acid residue could fundamentally alter the behavior of that protein. And so if we want to understand how proteins function in a complex network, we have to build models that somehow incorporate the functional aspects of this three-dimensional structure in a larger network context, and then are connected to prediction of some kind of cell fate or cell behavior. So this is really a multi-scale problem. Starting at the molecular scale, we have all-atom MD simulations we were talking about just a minute ago. At the highest level, we have sort of standard sort of reaction networks. Often these are modeled as simple differential equations or even Boolean variables that influence each other and can give rise to different steady states or kinetic transient behaviors. And rule-based modeling sort of sits in the middle and is trying to connect these two scales. So we want to be able to make an abstraction of a protein that considers the functional elements of that protein, binding to other proteins, post-translational modifications that can occur, and where these can occur in this context of many molecules that are similarly structured. So we have here RAS in the context of a growth factor signaling network. It interacts with many other proteins. It has modifications that modulate those interactions. And how do we build detailed models of the biochemistry? And I picked examples for my slides here that are talking about growth factor signaling, obviously, in the nervous system, in neurons. We have similarly complex. But the properties are very similar. The post-translational modifications that give rise to a lot of this complexity are shared features of both systems. So, so the basic idea, I don't know what that slide was. Oh, something, yeah, OK. What is this? I see. Yeah, please.
So I guess I don't really see how it's different than a reaction network. I mean, to me, it seems like a reaction network with more possible states for a given.
Right. It becomes impossible to. Right.
Can it be deconvolved into a reaction network again?
Yeah. Most of the time when we do simulations of these models, we simulate. We often use differential equations, which essentially amount to a mass action reaction network. But the fundamental representation as a set of interacting objects is what really differentiates rule-based from sort of standard chemical kinetic approaches. We'll explore that a little bit in the next couple of slides. But for example, the way that this network is drawn could imply hundreds or thousands of different biochemical species. But the interactions here, we can see that we have a relatively simple set of interactions. So it's much as when you have a force field that interacts between proteins that derives that you use as the basis for performing an MD simulation. Here we have binding interactions and catalytic interactions. There's a small set of them that can generate a very large state space potentially of different. So they would be different equations in my differential equation model.
So I just would say this is much more mechanistically accurate. To model a network with, say, one rate for each arrow is very coarse-grained.
Right.
And if you have a small system with not a lot of molecules in it, you won't be able to accurately simulate it without the kind of detail that Jim's talking about.
Yeah, we'll explore as we go. So basic idea, so the challenges of modeling these networks, proteins are multifunctional, so they typically are comprised of conserved domains and sites of post-translational modification. Phosphorylation is one of the most well-studied modifications, but there are many other types of post-translational modification that can modify the interactions of proteins. And so just this representation makes us think of proteins as complex digital objects that can exist in a very large state space and we want to understand what the rules are that govern the transitions in that state space. So there are many sites of binding, there are many sites of modification, and so representing what's known about the interactions requires handling of this problem of combinatorial complexity. So the idea of combinatorial complexity is when you have even a small set of multifunctional objects. So here I have the signaling molecules that are involved in early events in particular immunoreceptor signaling cascade. This is the high affinity receptor for IgE, also known as Fc-epsilon R1. So this was a model that led us to the development of BioNetGen. We had a system where we had 4 different interacting components. We had a handful of 6 or 7 rules that described the known biochemistry, and each of these rules was supported by biochemical measurements and experiments. And what we found is that when we went to make the reaction network corresponding to these simple interactions, there was this combinatorial explosion. So there are a total of, sorry for this being very small, but there are 354 different biochemical species that arise because of the combinatorics here. We'll do another combinatoric example in just one sec. And thousands of reactions. So originally, back in the day in Los Alamos, when we were sitting down trying to put together these models, we had like a committee of 5 people, and we divided up the equation space. So each person was responsible for writing, 50 or 60 equations, and we would meet and discuss them, and we would argue about factors of 2. And so we realized pretty quickly that we needed some automated way to generate these models and equations. And we'll come back to this issue of complexity. But the basic idea is that small number of components and interactions, there's a huge number of possible species and reactions. And you could argue, well, I could make a model that sort of sweeps all this under the rug, and that's possible. And I guess the 2 arguments I would give against that is, one, this represents primarily what we actually know about the biochemistry. If we make simpler models, we're making assumptions. Sometimes those assumptions are even hard to state in a concise way. In fact, there's another model of growth factor receptor signaling that we studied for many, many years. It took us a couple of years to kind of figure out exactly what the assumptions of the earlier simplified version of that. So there was a simple version of a model that had 30 or so equations, and the full expanded version had 300 equations. And the question for us was, well, what assumptions did they make that allowed them to have a simpler set of equations? Sort of something that you did mentally, sort of implicitly, and making those assumptions explicit actually is not so obvious sometimes. So in this particular case, we had no idea how to write down a simple set of equations to describe this system. Any simple set that we could come up with would violate certain basic observations that had already been made. So this sort of drove us to go beyond the sort of standard approaches. And we'll see other examples of that sort of thing. So in the growth factor signaling systems, many of them have these long cytoplasmic domains that have multiple tyrosine residues that are conserved through, at least in mammals. And they bind a bunch of different adapter proteins that are responsible for early events in growth factor receptor signaling. And the combinatorics become enormous because these receptors can dimerize. There are multiple isoforms of the receptors that can dimerize. So there's just a huge explosion of possible states of the dimer. And yet, it's very simple to write down one of these models in terms of the interactions between, each site can interact with a handful of different adapter molecules, and then there's a handful of sites. So we can write this as a pretty small set of rules. Maybe this model actually by the time you get done with all the different isoforms and all the different adapter types and so forth, it's actually a few hundred rules, but it's a lot smaller number of rules than the number of possible states that the rules generate. So again, this was driving us to develop methods that would allow us to simulate based on the rules for interactions between proteins rather than the number of combinations of protein interactions. So molecules are models as structured objects. So the basic foundation of rule-based modeling is that a multifunctional protein that has some complex three-dimensional structure and fold is abstractly represented as a molecule with a set of components, and each component can bind to other components, either in the same molecule or another molecule, and it can be modified. It can exist in multiple states. And those states could represent states of post-translational modification. They could represent conformational states. They can represent sort of any property of the molecule of interest in a more abstract way. And so it's a simple, rule-based modeling is really a simple object language, object-based modeling language for biochemical kinetics. And it's so simple, multiple people have invented rule-based modeling starting in the late 90s. And it's been, and since then it gets discovered every couple of years by different people who, are working in slightly different subfields. And the reason is this: it's sort of the most obvious next thing to do. If you're not going to represent each of these as A, B, and C, the next thing you might do is represent it as A with a couple of sites and B with a couple of sites and C. So that's where that comes from. So in BioNetGen parlance, we have molecules, components, and states, and we'll get into the syntax a bit more later. So we could represent, this is a kinase with multiple functional domains. This is a multi-subunit receptor. One way to represent a multi-subunit receptor, kind of a simplified way, is as the whole receptor complex as one molecule object that has different subunits. In this representation, this is assumed to be a fixed structure. There's always a fixed number of the same subunits. You could represent it in a different way as a complex that could allow the subunits to exchange, for example, and so forth. So that's the representation. And then the next step is to define rules. So rules define how molecules interact.
Can I just ask
Certainly.
It's too early to ask. What is the significance of the A underscore, B underscore, and G?
The underscore is just allowing you to continue the label. It's just a white space character.
But why is it A, B, and G?
Alpha, beta, and gamma subunits of this is alpha, beta, and gamma.
Okay.
Yeah. And so here we've abstracted. Gamma is actually a homodimer, disulfide linked. We represent it as one object with one ITAM. It actually has two ITAMs. There's an ITAM here. These ITAMs are phosphorylation motifs. Each ITAM actually has two phosphorylation sites. The beta subunit has a so-called non-canonical ITAM. I love all the biochemical literature. There's a non-canonical ITAM that has a third tyrosine that's thought to be inhibitory. So it's like a nested motif. You have, I think it's like an ITAM and an ITIM inside, so it can be inhibited. So there's clearly a complex logic, regulatory logic that's going on, that biochemistry, that evolution knows about and is using to construct circuits. And BioNetGen and other rule-based languages are sort of our first crude attempt to find a way to represent that logic and make models that are built on that logic. And I think when we fully understand it, first of all, we'll know a lot of principles about how signaling works better than we currently do. It's kind of pathetic how little we actually really understand about signaling. And what's more, we'll be able to use these kinds of tools to design new circuits. So, okay, rules define interactions of molecules. So, for example, this is the kind of thing that we know from reading the literature. Lin SH2 domain binds to phosphorylated tyrosine 218 on the beta subunit of Fc epsilon R1. So, and Lin is shown here, kind of in a cartoon form, binding, and we can represent that piece of information very concisely as a rule. Lin SH2 domain. So when we refer to a component in a molecule and and it doesn't have binding associated with it, it means we're assuming that it's unbound. So we refer to SH2, there's no exclamation mark after it. That means it's unbound. Similarly, the beta tyrosine 218 here has to be phosphorylated. So the tilde P represents the phosphorylation state of that subunit. It's referred to without an exclamation mark, so it's unbound. And it can bind then to form a complex. And the exclamation mark with an index is used to indicate the starting point of a bond. And when that exclamation mark with the same index occurs again, that's the end point of the bond. So there's a link between these two components. This one is phosphorylated. This one doesn't have an internal state. So we see there's a transformation here. We see a bidirectional arrow, much the same bidirectional arrow that occurs in M cell that we saw yesterday, showing that the reaction is reversible. So we have a forward and reverse rate constant associated with that. So by default, BioNetGen, let's see. So we see reactants, products. This is just standard reaction notation, the bond, and the rate law. BioNetGen syntax assumes mass action kinetics. These can be constants. They can also be more complex functions, which we may take advantage of later on in the lab. But for our purposes, we could just take them as constants. The key point here is we're operating on this principle of don't write, don't care. So here, when we defined what lin and Fc∈R1 molecules were in the previous slide, they had other elements that we're not referring to in the rule. And that's because we're assuming in writing the rule in this way that those other elements don't affect the kinetics of this interaction. So don't write, don't care. The elements that are not mentioned could be in any state. So that means that these rules can apply to many different complexes or many different species that contain a lin with a free SH2 domain or an Fc∈R1 with a free beta subunit. Yep.
So does that, what about like molecular inhibition or competitive binding or something like that? That does play into, right? I mean,
it could, yeah. So if we'll see, we actually in this model has a sort of form of competitive binding. There are actually two domains of lin that can bind competitively to this beta 218 subunit. But it's that, I mean, it doesn't actually, so there's a so-called unique domain of lin that binds to the beta subunit, and it basically includes this site. So we have a separate rule in the model for that, and the key thing is that once this site is bound, it no longer satisfies the requirement that the site be free, so it can't bind again. Okay, so that's how we would get competitive binding in a model, for example. On the other hand, if they were distinct sites, the binding would be non-competitive, and you would get a state that was bound. You could have 4 different possible binding states of that molecule then.
so what about like binding of the inhibitor introduces a conformational shift to the protein,
Mm-hmm.
and then now it can't bind this other protein. So theoretically, don't write, don't care would be like that reaction would still happen anyway, even if the inhibition was happening.
Right. So if we wrote it this way, and there was an inhibited, there was another site that was allosterically inhibiting the binding here, then if this rule would still allow, this would say the binding is independent of that state, so there's no inhibition. You'd have to. You'd have to specify the state that blocks this binding somehow. Yeah. There are ways in BioNetGen to using this energy representation where you can have basically allosteric interactions occur in a more narrowly defined way. But that's sort of an advanced topic that we won't. If you're interested, I can point you to the relevant materials about that. And I'll be right with you, James. I just want to finish my point, which is. Yeah, so this is really the simplest kind of assumption. Actually, here we assume that it has to be phosphorylated. The simplest kind of binding rule we would write would just have 2 free components that. The competition sort of comes in for these sites in that a site is only allowed to be bound once by default, and the logic of the language is set up really to make it so that in principle, you can make multiple binds to the same site, but the semantics will not. The language doesn't work very well if you do that. So so we typically have one binding site per physical interaction, and that covers a lot of ground in terms of making things work. And obviously, biochemistry is more complicated than this, but we had for most of the signaling pathways that we started modeling, those allosteric interactions are not well known. So we wanted to be able to write rules that incorporate that kind of modulation, and we can do that. We can make rules more complicated, but we didn't want to build in those assumptions into the language. So that's why it's the way that it is. Do you have a question?
Yeah. You could kind of write a great terms of equations of other states of clouds and mnemonic.
Yeah. You can make those arbitrarily complicated. There are certain semantic restrictions that are tricky, but we won't get to those cases, I don't think. Yeah.
Is there any sort of database or just example? Because I would imagine you'd use some of these reactive products like over and over and over again. They follow a lot of the same rules. So is there like a dictionary?
It would be lovely if somebody built a dictionary of rule-based terms. There are some resources for that. I can give you some links to that later on. But there's no sort of systematic unipro of rules. There should be. People have proposed that, and so far haven't gotten those efforts too far off the ground. But there are, for some specific models, there have been some papers published that provide sort of a library of rules for particular systems, like the FC_R1 system. That there's a paper about that you have.
So from rate law dynamics, I'm interpreting that there's a differential equation corresponding to each Markov, or a set of differential equations corresponding to each chain in the Markov space. Is that correct?
That's one way to interpret what this rule means. It's not the only way. So BioNetGen and rule-based languages support multiple semantics for the same model description. So we really separate how you simulate a model from how you define what the interactions are, and we'll see that. So the same rule could be used if you're probably familiar with this in the context of reaction networks, you know that you can do an ODE simulation or a Gillespie simulation of the same system, right? So the same way. We have here, we can interpret this as a set of differential equations. We can interpret it as a set of processes in a Markov chain, like with Gillespie. We can also interpret this as a set of transformations on objects and do an individual-based simulation, which lets us avoid generating networks, which we'll see in a few more slides. So good questions, thank you. This just says in words what I said before, that the reaction rate is determined by mass action kinetics. So the forward rate would be the product of this variable KPL times the concentration of free Lin SH2 sites times the concentration of free FC_R1 BY sites. And rules can be more complicated than this, both in terms of the context, the properties that the reactants have to satisfy, the nature of the transformations that take place, the nature of the rate law constants. Those can be functions. And we can also restrict rules to only apply in certain circumstances. But we won't get into the details just at the moment. So essentially, we have a workflow for rule-based modeling that looks like this. We build up an interaction network comprised of this signaling molecules of interest, sites of interaction with other signaling molecules of interest, sites of post-translational modification that modify those interactions. These are defined as the objects and rules that go into a model in the BioNetGen language description. And then that model can either be interpreted as a reaction network, and in this case, we're going to iteratively apply the rules to the molecules, the base molecules in the system, and generate a reaction network. That's all the possible species that can arise in that network and all the different reactions that can occur among them. And then we can do sort of standard things with the reaction network, simulate a set of ODEs, perform a stochastic simulation, This is going to give us time courses for the different model states as a function of time. We typically define specific sums over those states to get aggregate variable outputs, much as you did with molecule counts in worlds, for example, yesterday in the lab. So we can look at time courses for the model for a given reaction network for a given set of parameters. And then there's another. So as I mentioned, this combinatorial explosion problem is not really fully circumvented by this process, because this reaction network, as I said, could have 10 to the n states where n can be a very large number. And at some point, you run out of memory to put all those states and reactions. And that point is typically in the neighborhood of between 10 and 100,000 reactions. Things start to get pretty intractable. And so in those cases, we can simulate a model by what we call direct methods. And direct here just means that we're using the rules themselves to drive a simulation rather than expanding to a reaction network first. And we can perform a stochastic simulation using that method that scales very well as a function of the network size. So a little bit more to delve into that, just a little bit more detail here. So if we have a model specification with an indirect simulation method, we would generate a network. We get that reaction network, and then we pass that reaction network to a reaction network simulator. And for those of you who have built models of reaction kinetic systems, you may have used many of these tools, things like COPASI. SBML is a standard format for these kinds of reaction networks. There's lots of tools that let you perform simulations of reaction networks. We have some built into BioNetGen, a good ODE solver, and this Gillespie's direct method. But in cases where the model specification results in a combinatorial explosion of species, and you get thousands or hundreds of thousands, it may be better to perform a simulation where you instantiate the specific molecules that are in a given cell, for example. So you may simulate 100,000 molecules, or a couple of 100,000 molecules, using a kinetic Monte Carlo simulator. The one that we have built, that we have associated with BioNetGen, is called NFSim. And that allows you to perform a simulation without doing this network expansion. We call that network free. And yeah, it says that. And the basic idea is that if you're building a model, for example, of our favorite Fc epsilon R1 signaling pathway, typically we want to include more model components and interactions as we're sort of building up that model. And so as we're doing that, we're increasing the size of the potential reaction network that's implied by those species and reactions. So here we're looking at different models where the network size is increasing over several orders of magnitude. And then how does the runtime of that model scale with the network size? And what happens is that if you have to generate networks, so here's the ODE simulation, most of the time that's spent here is spent in generating the reaction network. And then and the simulation is also scaling pretty poorly with network size, typically approximately quadratic for the differential equation system. And so what happens is you rapidly run out of resources to simulate your models if you use this sort of indirect approaches. Yeah, question.
Yeah, so I mean, I said the kinetic Monte Carlo reduces the generation size of the graph, but I don't see how you're avoiding having to specify the kinetics and actual ODEs for every single combinatorial possibility if that's reached.
You basically are figuring out what the appropriate kinetic transformations are as you encounter the states. And you can do that in a very efficient way for a particular collection of individual particles or individual objects in your system.
So you don't use an ODE, it's an algorithm. So an ODE gives you an average of a whole lot of things. You just use an algorithm. You follow the rule, you follow the rule.
Do I have a picture? I don't have.
It's different. There's no ODE about it.
You had a system, you're sampling from its solution set, and your distribution attributes that you get. I have a question. I didn't understand network free.
I understand. It's tricky. So do I don't have, well, we can go back here for a sec. I have some slides that go into this in more detail. And don't worry, and it is a concept that it's a little bit subtle, and people it tends to take a while to kind of get one's mind fully around it. But the basic idea is that instead of, so when we do ODE simulations, we're basically, mass action kinetics was a theory that was developed to describe what happens in a reactor, right? Where you have, Avogadro's number of molecules of different, or species of different types, right? And you have a small number of different types, but you have lots and lots of molecules. In a cell, you're somewhere in between, where you may have many, many different kinds of states of proteins, and other complexes, and complexes of proteins or other biomolecules in the cell, but relatively small numbers of molecules. So in some cases, it can become more efficient to actually build a simulation in which you explicitly model each of the individual proteins as a separate object, and then use rules to determine how to change the states of those proteins and how to form, for example, bonds between different proteins to form complexes.
This kind of reminds me of M-cell. But am I wrong?
It's a little bit, there's, yeah.
It is exactly what M-cell does.
There's one extra step in which the rules can actually query specific properties of these objects that are represented explicitly as different states, whereas the species in a sort of standard M-cell model just have a label, and that defines everything about the species.
So, and we made an experimental version of M-cell where we added some of that to M-cell, but it wasn't expressed with the bioNetGen's language. One of Jim's students, Jose Papia, who just presented his thesis last year, successfully merged NFSim into M-cell so that we would have the bioNetGen language and get exactly this. And that's still experimental.
So the key idea here is that these little colored boxes on the left here, these are the rules. So you have a handful of rules. Even in the most complicated EGFR network model, this is like 10 to 100 rules. These are the event generators, and they're basically querying properties of the system that's being simulated and telling you what events can fire at a given time. So this is a standard, description of a discrete event simulator, and the key thing is that we never try to enumerate all the different possible states that each of these molecules that are queried by these rules could be in. So there's always a fixed number of event generators. Okay, if you built a simulation based on a reaction network, say a Gillespie simulation, there the event generators are all the different reactions, and you'd have to figure out what all the different possible reactions were explicitly in advance to do that. Okay.
So here it's a network of rules, but there it's a network of objects.
Of reactions. Yeah.
The rules and reactions. Also,
They're not really. They're not. I mean, you could.
Different syntax or a different way of expressing the same idea. One way is explicit and blows everything out into every possible state.
Which becomes computer intensive, and that's the reason for not doing it all the time.
But another way to express it is as rules, right? And the rules are like little algorithms that if you expand them locally, expand out into a small local network.
but locally, I thought there was. So
Locally, state by state. A species knows that it's in this state.
maybe it'll help
And
find
these are the adjacent, and this is the rule that applies to me, that indicates these are the possible state transitions I can do. And those are the only possible ones that I can do. And those are the only ones I have to consider. And then I transition to the desired state. And then now I'm in a new state where a new rule applies, but I don't have to deal with that until that happens, right?
How does an event get turned into one event?
Okay, so let's just think about A binding to B. So let's just assume we have the simplest kind of rule that says A binds to B means that A has to have a B binding site that's free, and B has to have an A binding site that's free. So the rule at the start of the simulation will query the box, the set of objects, and find all the A's that have a free site and all the B's that have a free site, and it makes a list of both of those things. Okay, and so each of these rules does something similar. It finds all the possible individuals in the system that can react under its defined rule, and then at a given time, at a given step in the simulation, you have lists of all the possible reactants. You pick one of these reactions to fire based on a weighted, basically sampling the propensities. So the higher the rate, the higher the propensity of the rule to fire, the more likely it is to fire.
Or the more reactants.
Yeah, exactly. The more reactants. So for each reactant combination that can occur in the system, adds another K reaction propensity amount, right? So the number of combinations basically weights all of these propensities, and so you sample that way. And then when you fire, when the rule fires, it makes a change, and this is what Tom is referring to as the local configuration. You make the bond between A and B. Now you have to update your list, but you only have to update the list for a small number of objects. So it's sort of standard. For people who've done Monte Carlo simulations, sort of standard things apply. Again, it took us a couple of years from the time when we had. So there was a period of time when we didn't have this method, where we had the general rule-based syntax. You can imagine we were like, oh cool, we can now describe all the interactions of the growth factor receptor signaling, the growth factor receptor tyrosines with adapter molecules as rules, and so we frantically wrote down all the rules for those, and then we said, well, damn, that now we have and I remember there was a time when we had, BioNetGen is written in Perl. It was a prototype written in Perl that we intended to transition and still hasn't happened. But so we had BioNetGen, we had all these rules, and we would start to generate reaction networks and where that would supersede the memory. And this was back when we had only 32-bit machines. Then 64-bit machines came along right at that time. So this was like 2003 or 4, maybe 2005. So I was like, oh cool, we're going to get around this problem with 64-bit memory addressing, right? Because we can get it, instead of 2 gigabytes, we can have 4 or 10 in memory. And so that was our idea. Well, that doesn't go, then you just add one more tyrosine and then boom, it's gone. So that's what happened. That was literally the genesis. First we were doing this and we thought this, because when you have the ODEs, it's much faster, right? Or even Gillespie is very fast compared to, well, there's a crossover, but it was thought to be very fast compared to doing something like this. But we quickly realized we needed this to go beyond simple models. So, and nowadays it's become quite fashionable to think about polymers forming in signaling complexes. So there have been some really blockbuster papers showing that that some of the components that were just thought to be simple adapter molecules can actually form polymers of receptor molecules and other transmembrane proteins. Of course, people who study postsynaptic density knew about this for a long time, but you have the possibility to form these very extended aggregates. The number of possible chemical species in that system is infinite, but you can start to describe the chemical kinetics of such a system with simple rules like this, but you need a way to simulate that, and that's also what these direct methods do.
Non-postsynaptic density adapters that polymerize.
LAT, linker for activation of T cells. Been a series. There was Ron Vale had a Science or Nature, I can't remember, in the last 2016. The first Mike Levine at is it Mike? Not Mike. Levine at UT Southwestern. What's his first name? It's M. I can't remember. He had a paper in Nature in 2012 about WASP and NCK that polymerize. And I guarantee you're going to hear a lot more about it because it's all the same molecule. Sorry? Schick does not polymerize, but GRAB2 and SOS can form a 2-to-1 complex where you have 2 GRAB2s bound to the sauce, because sauce has these proline-rich domains, and that bind to the GRAB2SH3s. And those linkers can link all kinds of stuff, because GRAB2 is promiscuous binding to phosphotyrosine of many different cytoplasmic domains. And so it can cross-link. And we've only started to scratch the surface of these complexes. It's true in the innate immune system, too. All these, the death domain and the TIR domain, and there's a whole bunch of other ones. And there's a woman at Harvard who's looked at this in the innate immune complexes. Min Wu, maybe? I think. It's a big thing, and it's emerging, and these methods are very important to be able to deal with those problems. And it's just the tip of so you need this. This is all non-spatial, whereas this is all assuming that we have well-mixed compartments at least. But when you have polymers forming, then the molecular scale becomes a macroscopic scale. You have, and space has to get considered in some way. So that's sort of the current frontier of making simulators that can handle polymers, that can handle extended space, and explicitly have concentration, or the concentrations of molecules varying over space. And do that in an efficient way that scales. So that's a major frontier of investigation, I would say. Okay, so the basic workflow to couple systems models with biology could look something like this. This is how, in my group, this is the kind of thing that we're trying to do. We build biochemical models using rule-based approaches typically. We then need to calibrate our models to at least match experimental observations. Here, we're trying to calibrate models to quantitative data, but in some cases, we're just happy if we can find parameters for the models that match qualitative observations. But and here we're using, I'll talk about it a little bit, Bayesian approaches where we get different sets of parameters under which the model can match experimental observations. And then once we're satisfied that our model is a reasonable representation of reality, we start to analyze the model to find interesting behaviors. And hopefully those behaviors will be robust. So here we've used a Bayesian method that allows us to estimate how uncertain our model predictions are. And we see that there's a robust behavior here of a sharp threshold in signaling with respect to the dose, the input stimulation of the system. There's some outputs that that switch off very sharply at a particular dose. That's a robust observation. Doesn't matter which parameter sets we use. If it matches the experimental data, we get this kind of behavior. So that suggests we should be able to find such a switch experimentally. So we didn't do some experiment, convince our collaborator to do some experiments that hopefully show that this is true. If it's not true, then we know we've got something wrong with the model and we have to fix it, and we go back and do that. So we keep, this is why I call closed loop systems biology. We're constantly making predictions and getting feedback from experiments and trying to refine models. Yep.
Is there any work on generating the actual graphical models from the data in an unsupervised manner?
There's a little bit of
Exploring the space of connections, I guess.
Yeah, there's a couple for discovery of this sort. There's just a little bit. So when I say discovery, I mean you have some experimental data and you want to find what's the true model. That's one way. There are some. There's a nice system called BioJazz that is doing that for evolution of models, where they're trying to evolve model structure to meet some kind of engineering objective, like this, for example. How do you make a switch out of these components? What are the, sort of, and find me parameters and model structures that give me switches.
Integrated pathway analysis is similar to that approach. Taking a RNA-seq data and pumping that through, looking at everything that changed, building a model from that.
I think the key principle here, and learning models is, so people have done this also. The sort of simplest kind of model learning that people have done on signaling data is like partial least squares kinds of model learning. and there have been other sort of similar flavors of learning models from data for signaling. But here, what distinguishes this kind of using rule-based system to do it is that you really have biochemical mechanism underneath as opposed to some regression model or something much simpler. So there's a lot of nonlinear feedback here, for example. There's a much richer structure of behaviors potentially here than in what people have mostly done up to now, with the exception of the BioJazz is an example. And there's probably, if you look in the literature, there's probably some people who have worked on that, but nothing like major that I'm aware of. And I think that's a big challenge problem for the field. So go for it. OK, so that's this is like science. This is like science with biology inside. So most biology is done with this part, and instead of this stuff out here that's systematic and quantitative, we do it in our heads. So I show. I don't have this slide in this particular presentation, but I often show it's a cat. The cat is trying to figure out what's going on with the. This is the diagram that the experiment is on, and then they make a prediction, and then they go and test it. But our ability to, as soon as these are complex feedback circuits, our intuition is gone, and you shouldn't do that without a model underneath. And it's really fun. I mean, there's no reason to, because the tools that build these models are pretty easy to use, and you can play around and discover interesting things quite easily once you have a little bit of knowledge about how to do it. OK, so this is a very complicated diagram just to convince you that BioNetGen has a lot of stuff going on in it. So the core of BioNetGen is sort of represented on the inner parts here, and then as you go out, it connects to all kinds of other tools. So we have a very, as I mentioned, essentially, instead of ODEs and reaction network models, BioNetGen lets you have structured object models. You can imagine that would be very useful to connect to all kinds of other simulation and analysis platforms, and that's basically what I'm showing here. We have an application note that came out last year that describes some of the newer capabilities of BioNetGen. There are a lot of, once we make a model that we generate, where we generate its network, we can export models in a variety of different formats. The systems biology markup language is one. For people who use MATLAB, you can export the model as MATLAB ODEs. You can export the model as a MEX file that you can compile and make run really fast. We can export models to MCell, which hopefully we'll show in lab. We've been having certain technical issues with that recently that are temporary, but may prevent us from going into much depth on that. And we can export models in a visual format that we'll demonstrate today a little bit later on. So I think I'll leave that. Just can go into more detail another time. The question was asked a little bit earlier. Are there libraries of models? Not exactly, but we have lots. We've been trying to put together examples from the literature of rule-based models where people have used BioNetGen to build complex models of systems, and hopefully have provided the files for you as supplemental material. And we're trying to keep this up to date, but we're already behind, I think. And a lot of other tools have incorporated BioNetGen. So PySB is one of them. I don't know, some of you may have used that to do simulations. PySB uses BioNetGen underneath to take a model description and turn it into a reaction network. BioNetFit is a tool that's developed by one of my collaborators who's at Los Alamos to do parameter estimation on rule-based models. In particular, BioNetFit can be used to do parameter estimation on NFSIM models, so network-free models in a highly parallel fashion. So that could be a very useful tool, and that's under active development. They're improving the capabilities all the time. So that's good for, that tool is very good for hardcore project use. There's an emerging standard for rule-based models that's a part of the SPML standard called Multi. So currently, it is possible to export a model from BioNetGen and have it import into Simune. That's kind of an experimental capability, but ultimately you should be able to build these kinds of interaction-based, rule-based models and simulate them in a variety of different tools. BioNetGen is already built into the virtual cell, so you can build models visually using the virtual cell interface and simulate them in virtual cell as ODEs, as Gillespie simulation, or you can also do PDE simulations of rule-based models, although that's a little bit trickier. And then there's some other tools as well. So here's the virtual cell interface. So if you like, you'll see today we'll basically deal with the text-based interface to BioNetGen. If you prefer visual interfaces, the virtual cell one is pretty nice. Although it's still basically text rules as instead of text. Now we have a kind of a eye candy based. It's not text, but the translation between this and the text base is pretty straightforward. But it looks better, and it's a nice. This is a nice interface for managing models. Okay, so just a quick summary. Why would we use rules? They're a concise and precise way to represent biochemical knowledge, and rules are very simple when interactions between when the different binding sites on a protein are largely acting independently, then you can write rules in a very concise way. When they're strongly coupled, then rules get more complicated, but so does the state space of the model. So you just have messy reality interfering. And if you have problems where there is a lot of coupling, talk to me, because we have ways of managing that go beyond the sort of simple rule-based syntax. They're flexible with respect to simulation methods, so we can, the rule-based model is just a description of the interactions of the molecules. It can be simulated in many different ways, and we'll look at some of them in the labs today, but there are others. And those involve slightly different workflows involving different tools, and as you've already seen from yesterday, when you try to couple different complex tools together, there are glitches. But it's all work in progress. It can be done, and if you have an application that needs it, then we're happy to facilitate that. And this idea of rule libraries, it's an obvious thing that I think should be a driving force for the field. The fact is, and it's frustrating to think about. Our current view on signal transduction is biologist-driven. My system is special, and I have a very particular favorite molecule that it does this thing. But in fact, there's a sort of a universal set of molecule, and the interactions in molecules are reused from system to system. So if we had a universal library, a uniprot of rules, we could build models for any cell of interest, more or less. And then we could even simulate them using network-free methods. Figuring out what it all means is still very much a work in progress. But there's a universality in signal transduction that is, I would argue, is just not being probed properly by the current state of science. You're going to express that it's not different. I mean,
that, for example, in the synapse, there's an enormous amount of specialization.
That's true. That's true.
It's just not anywhere else. I mean, there may be.
Yeah, I agree. So there's definitely, I agree with completely, and I'm trying to be provocative, so I'm glad I provoked a reaction. There's a core that's universal, and then there are leaves that are specialized. So isn't that how evolution works? I mean,
yes,
you have this nice common set of components, and they can do all kinds of different things. And even those same components are rewired to do different things in different cells, and we don't really. Yeah, RAS.
Very important in the synapse.
Right.
And everywhere else.
Right, and CAMK2 too is probably
It's more specialized.
Isn't part of it?
What?
It drives counter-side hypertrophy, so at least in two.
Okay.
And if it's a good, and if it's.
Usually overexpressed in the so it functions different. It's like 2% of protein.
That's interesting too. So we could have a lot of fun with these things. But So I think that when we start to think about models in these ways, there's a lot of possible. It changes our view of what signaling is, and I think that's useful. And we've been working a lot on trying to come up with ways to visualize rule-based models, because it's very easy to write a whole bunch of interactions and not be able to see what those are. And so the contact map is a very nice, simple way to see what a set of rules does. I mean, this is encompassing something like 15 different rules, and the text form of these is very complex, but you can see at a glance what's in the model. But if you want to be able to understand at a more functional level what's going on in a model, you need to be able to look at the consequences of multiple rules at the same time, and that's what the visual representations are for. This was alluded to already by Tom. We're trying to couple rule-based simulations with spatial stochastic simulations using MCell, and my former student Jose has developed an experimental tool called MCell-R that does exactly that. So it's MCell with rules, and we're working to couple the two simulators. And that's part of the motivation for the workshop and why we teach both tools.
We kind of got it to work last week.
Yeah.
But that's a little too
it works, but it works in,
to cell blender.
Yeah.
And so from the user interface point of view, it's pretty nice. But the back end sort of works, but there's some tests that fail. So we're not ready to really recommend that your mortals use it.
But if you're starting now and you're interested in some complex biochemistry, you could start playing with the well-mixed versions of those models, and be confident that in the coming 6 months or so, it would be very possible to do simulations of that in a spatial context. Really less than 6 months. I mean, and I would encourage you, if you're interested in doing that sort of thing, get in touch with us, because we can help you to make sure that your tools work.
You need to do some beta testing. See.
This is just the teaser for stuff that's coming later. Hopefully this afternoon, Atomizer will talk about how to get models that are in sort of standard reaction format and convert them into rules. And that's great for even just seeing what's inside of a model. Why do I have all this? Oh, I see. Visualizing models. So if you have typical rule-based model, it has this pretty scary looking code. Each model you can figure out what each rule does in the model, but figuring out what they do on a larger scale is difficult. And so, so we've developed techniques to visualize individual rules and then break the rule visualizations into pieces and make large graphs. Oh, yeah. Somehow the slides here got a little bit backwards. Let's see if I have a picture of that. I don't have.
Can you stop on the rules or code slide for a second?
This one?
Oh, just part of the model we were trying to make. The old syntax was.
So I mean,
Code even nastier than this.
the teaser for this problem is that this is the typical format that you have most rule-based models in. You can get a contact map that shows what the basic molecules and interactions are, but to understand, having some view of the architecture of the system like this was a hand-drawn diagram, is tricky, and we're working on methods that give you. I just don't have the picture. I'm sorry. I keep looking for it. Basically, we can generate a diagram that looks very much like that just from the rule set itself, and a set of using the visualization methods that we'll talk about in the lab later. Will show you how to make diagrams of complex rule sets. And let's see, I mentioned the model calibrations, P-Tempest tool, that if we have data and a model and we want to calibrate the data to the model, and the model to the data rather, to find sets of parameters where the model fits the data, we use this tool called P-Tempest, and Sanjana will talk about that this afternoon. And with that, we're sort of ready to go to the next stage, but I'm happy to take questions. Yeah.
So if you have one of those like architectures that you showed, like a lot of times we don't know exactly that each of those pieces are true, like the sign
Mhm.
Is it easy enough to just like X that out and then try the simulations in different ways?
Yeah, it's very easy to to modify the structure of the model. Typically, it's easier than well, depends on it depends on the particular what the particular thing that you want to do and how your model is encoded. But I mean, it would be very easy, for example, to just set ULK to 0 in the model. You could do that in any reaction network model. If you wanted to ablate certain interactions in the model, that's usually very easy to do in the rule-based form because that's explicitly encoded as a rule, and you could just basically comment that rule out or set the rate constant for it to be 0, and then it would be gone.
Is there like ways to put in probability on those?
Reverse reaction kind of do that. You can also rate constant.
But the Markov chain would, you have chains, you could have separate chains where you don't have USP1. You could have submodels, and these are rules, right? You could make a rule which doesn't include this USP1. But you have to write that a priori, I guess.
Yeah, I mean, yeah, you
This is a separate model.
yeah, typically, yeah, so typically what you would do is, yeah, you'll see in RuleBender it will be very easy to just comment it out or to set the value to be a much smaller value. And I think when you say probability, I think what you mean is that that's a measure of the uncertainty about whether that interaction is there. How would you incorporate that into the process? You could basically try to convert using Bayesian estimation, parameter estimation. You could convert that into a prior, like what's your expectation that rule exists. And we do that. We haven't done that extensively in the context of like a kinetic rule, saying if ULK1, if this interaction is present or not, like if it binds to something. But what you can do is you can make a prior that's centered at 0. So the most probable value of the most likely value of that parameter is 0, but it could have a non-zero value. And what will happen is if the data forces the model, if the fit isn't very good with the 0 value, the estimation process will force the parameter to a non-zero value. And so Bayesian estimation is great for that sort of thing. And that's something that has not been applied. I think its full potential has not been really realized for parameter estimation, but I don't remember. See if P. Tempest, I have this slide here. One of the things that we're investigating a lot now is using regularization strategies. Doesn't say here, but you can, if you formulate the model in the right way, you can regularize on parameters. So which means that you bias the estimator to give you a 0 value of that parameter, basically to remove that, whatever that process is from the model. So you only include that process if the data is driving that. That gives you sort of this, that sort of pushes you to find the simplest possible models. And that's a huge area of research now in machine learning community of different strategies to do regularization and basically model simplification in a systematic way. There's lots of, so there's lasso L1 norm, there's elastic net regression, there's all kinds of strategies for doing that, and different pros and cons, and get into sort of almost deep philosophical issues like, we're trying to fit a model. Should we, in lasso and maybe these regularization strategies, try to fit the simplest model? Is that what nature does? Does nature fit the simplest model? Maybe not. Maybe nature is perfectly happy with a solution where all the values of the parameters are non-zero, and it uses all these mechanisms because like some other thing keeps them to be non-zero. So we don't know what the best strategy is, but I can tell you that some of the regularization strategies can be quite useful for getting reasonable models from data.
Slides, can we download that?
Yeah, basically, it should be posted onto the agenda. I may have added a few.
The first set of slides was, but this.
I added some, and it must not have updated the. I put this on Dropbox, but I can make sure. There's no problem to get these to you.
Because this is very useful.
Sure. sure, yeah.
Can we regularize in a general way to say these are things that we expect in a biological network? In the same way that you could say an electron density map from electron microscopy is kind of smooth, that you could say there's something about a set of rules or a network that we want to enforce when we regularize it, when we try.
Yeah.
Like,
You can use whatever, you could enforce whatever strategies you like on that, definitely. Would not be, I don't think it would be hard. The code, and Sanjana will show, we're not going to do a lab demo with pTempest because it uses MATLAB, and not everybody, we don't have it on the lab machines here, and not everybody has it. We recognize that's a deficiency for the people who have strong opinions about that, but we haven't gotten around to translating that code into Python, which is what we would like to do.
All of this requires money, which requires NIH to function.
Right. Well, if you're a university, like
It's a big
here
requirement.
at Pitt, it requires minimal money because we have a site license. So it's like
But you
No,
have
for
to,
MATLAB, no, but the
you,
translation
you
part.
have to buy people.
yeah.
That's what I
No,
mean.
it's true.
You
You need
apologize
absolutely.
for not having these things done.
Oh,
You
sure,
have
sure.
to buy people.
Oh, I know. I
So don't apologize. Write your congressman.
yeah. So my student Justin, who wrote the pTempest code, probably was like the least likely person to write the code in MATLAB, but it was fast and it ran on the cluster they had. So like he just did it. And there's Bayesian parameter estimation code for Python as well that could be easily adapted. There's nothing specific about rule-based modeling to general strategies for doing Bayesian parameter estimation, but it can be cleverly applied to rule-based models in a variety of different ways. And yeah, be happy to talk about those offline too. Other questions? Yeah.
I have a related question that relates to about so typical like model that you built on top of biological. How robust are models that are built in this structure? Because there are general methods to test model robustness.
No, wait, but probably.